All right. So going all the way back to the beginning of the animals, what I want you to think about in your studying is to go from least advanced to most advanced. How do we go from this crappy sponge thing all the way to human, right? Even though humans aren't necessarily, quote unquote, the most advanced, but they're very recently evolved. So how do you go from a sponge to a fully formed advanced animal? That's what you want to really think about. That's the scaffold that will put all these individual like morphological features into place. Okay, and I'll really highlight that as we go on. So let's start. Blah 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 blah. Okay, so this first lecture was talking about sponges and diploblastic animals. So clearly the most simple of these animals, right? If you just look at the tree of life, you go from sponges, they come off very early, the most early, and then the diploblasts come out a little bit less early, and then everything else is more recent, right? So the further they diverge off, it means their ancestor that shared with us was slightly simpler. So we'll start out with symmetry. Let's just go over, like, what is the point of symmetry? What does symmetry get you? It gets you better locomotion, but the entire point of symmetry, just right from the start, going from a sponge to a coral, is symmetry gives you the understanding that these individual cells have come together to make a specialized structure, a tissue, right? So sponges have no tissue, so they're not symmetrical. Sponges have no tissue, so they don't have any germ layers either. So they kind of go hand in hand to some degree. The next level of symmetry is the radial symmetry, and the next level after that is bilateral. So you go from most simple to most advanced. Why is radial symmetry simpler than bilateral symmetry?
It doesn't give the organism directionality. Like
Right. Right. So while in the radial symmetric organism you have very specific tissues and very specific structures, it can't move around backwards and forward like a beetle can, right? This anemone can't move like a beetle can. The beetle, when it's front to back, when it's got front to back symmetry, it can move forward really fast. And what that also allows you to do is it gets you, so number one, it lets you move faster. Number two, it lets you develop specialized structures, right? If you have a front, then you might as well throw your brain in the front, you might as well throw your eyes in the front. You throw a bunch of these specialized organs that let you interact with your environment really well in the front of your body, right? And specialization breeds into advancement of function, right? That makes sense to everybody? So remember that. Okay, so in terms of symmetry, the ones that you need to know for the no symmetry, sponges, right? No germ layers, no technical germ layers, so no symmetry. Can you guys fill out the Professor Hoppen, please? All right. Thanks. So then that next level over, in the radially symmetric organisms, which organisms do you need to know that are radially symmetric? So we're just going to go over and review the names of them. Which ones? Number one are the cnidarians, right? Phyla Cnidaria. And then what's another phyla that has radial symmetry? Ctenophores, which are related to cnidarians. They're both bilaterally, or they both have two germ cell layers. And then finally, there's one exception all the way in the triploblasts that has radial symmetry that you need to know, and which one is that? The starfish, right? Echinoderms can be radial symmetric, except for the sea cucumbers. Okay? So everything else, everything that is triploblastic except for the echinoderms are bilaterally symmetric. So once again, sponges, no symmetry, ctenophores, echinoderms, and cnidarians are radially symmetric, and then bilateral symmetric is everything that has three germ cell layers except the echinoderms. Great. Okay, so germ layers. We've gone over this a billion times. Endoderm, mesoderm, ectoderm. Endoderm makes up what? Or organs, especially the inside of the GI tract. Right? So your digestive tube, that is your endoderm. Mesoderm makes up what? Muscles, skeleton, blood, connective tissue. And then your ectoderm makes up your skin, and then what else? Your nerves, right? Your nerves, they kind of go inwards from your skin. All right. So going back actually real quick, body form symmetry goes along with blasty, right? Remember, the sponges have no blasty. They're the simplest, so they have no blasty and they have no germ layers. The radially symmetric organisms, they have 2 germ cell layers, mostly, except for the echinoderms, and they're radial. And then for the bilaterians, they're all triple blastic. Right? So it's good to take those 2 concepts and merge them together like that. Okay, so we'll get back into the germ layers in a little bit when we go over the coelomate organisms. The germ cell layers, we already went over that. Remember, in the 2 germ cell layer groups, which you need to know, which are the ctenophores and the cnidarians, they have this bullshit in the middle, which is this hydrostatic skeleton, which kind of acts like muscle. Just remember that. And that is called the mesoglia. Just remember that. Mesoglia, that's going to be real important, I think. If I were to write questions, that's something I would write for sure. So that's in between the endo and the ectoderm in the cnidarians and the ctenophores. Mesoglia. Okay. Okay. So sponges are very, very simple. They kind of just act like individual cells that are moshed together with like this external cell matrix. That's kind of what they are, and that's why they have no true tissues and they have no true symmetry. All right, they're suspension feeders. They just feed off the water column, and the way they do that is through this cell type. This is going to be very, very important. You have a couple of cell types you got to remember. This is the most important one. The choanocytes, the collar cells. The reason they're called collar cells is because they have that collar, which is a net. So they have the cilia that beat, right, or the flagella that beat water through that net, and that net collects all the food. Simple. So let's go through the structure of the sponge. You have the pores on the outside, the ostia, which take in water, and that water goes through the sponge, goes on the inside, which is this chamber right here, and goes out through the osculum. Osculum is the big pore on the top where it goes out of. The ostia are the little things that line the walls that the water goes into. Okay? And all that water movement is facilitated by the choanocytes. And these are the food collecting cells that also move water in the sponges. Okay? You'll have a bunch of other cell types. You have the porocytes, the amoebocytes, which move the food around, kind of distribute nutrients and stuff. That's all in your lab notebook, and that will be in the notes, or I think that is in the notes. So I wouldn't worry too much about those. Study those for sure. The main thing is knowing the choanocytes. If you have to know one, know that. All right. And as we go from asconoid to synconoid to leuconoid, those are the 3 sponge types. Just remember everything in red is covered in those food cells, the ones that take in food. So the more red that you have, the more surface area that you have per water movement, right? And so as you get more and more advanced, the more advanced sponges will have a more advanced body structure, which is the leuconoids. So the calcareous sponges are the simplest. They come off of the sponge. I guess they don't have the tree of life, but the calcareous sponges, they pop off really early on. So they can be all of these guys, right? But for the demosponges and the glass sponges, they are only, because they're more advanced, they're only leuconoid, okay? And just know, as you get more to the right, you get fatter and you get more surface area. The way the water moves from the outside to the inside is a lot more windy. Okay. So do you guys remember? So this is important. You need to know the classes of sponges. These kinds of differentiations. When you see one slide that has like multiple classes and some like purposeful like distinguishing features between them, you'll need to know the classes. Okay, so 3 classes: the Calcarea, Demospongia, and the Hextolinids. Right? How are they different again? Do you guys remember? They're spicules. Yeah, their skeletal features are different. On the left, the Calcarea sponges, they're made out of calcium carbonate. These two, they're made out of glass. The demosponges and the glass sponges are made out of glass. In addition, the demosponges have one other skeletal feature called spungen. Spungen, that's what makes them like real spongy. Okay. And how we differentiated them is we dip them in acid and base, right? In the ones that have glass spicules, remember the spicules are those like little spines inside the sponge. When you dip them in acid and or base, they stay the same. All the shit dissolves, all the body stuff dissolves and gets gushy, and all the spicules stay there, right? In the Calcarea sponges, in acid, those disappear. Okay. Simple? We've already went over this a bunch of times. Just make sure, does anyone not understand this experimental setup? Make sure you know this. This kind of shit is like very elegant, so it's elegant and it ties in a bunch of features, so it'll be on the test, probably, for sure. Okay, spicules, we went over that. All right, cnidarians. So first group of cnidarians, the first phyla are the ctenophores. They look like jellyfish, but they don't act like jellyfish. So a couple of different features of them that jellyfish don't have. Number one, in the true cnidarians, they just have a chamber. They have they act like a bowl, right? Everything that comes in comes back out the same hole. Their mouth acts like their butt. These guys, they have a true digestive tract that goes all the way through the animal. Okay, that's number one. Number two, the way they move, the jellyfish, they move with the pulsations of their bell. These guys move with cilia that line their body. Cool? And that's why they're called comb jellies. They have these combs of cilia that beat backwards that allow them to move very smoothly through the water. And anything, remember, we don't have jellyfish in the lab, so anything that is live and moving around that looks like this is going to be a comb jelly, not a jellyfish. They have 2 tentacles that don't have the stinging cells. They don't have stinging cells. The cnidarians do have stinging cells, so that's another different thing. The last feature I would probably remember is on their butt, they have this thing called statocyst, which basically acts like an inner ear. It's an organ that senses gravity that allows it to right itself up. Okay, that's just something you just got to memorize. Okay. Blah, 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 blah. Okay. So now you're at the cnidaria level, right? This is a phyla that includes. So the 4 classes that you'll need to know are the scyphozoans, which are the jellyfish, the anthozoans, which are the anemones and the corals, the hydrozoans, which is the hydra, and what was the last? I think that's it. I don't think you need to know box jellies. Never mind. So that's 3 things to remember. Actually, both comb jellies and these guys have the mesoglia, but what's the mesoglia again? It's the hydrostatic skeleton, and it's the jelly that is in between the 2 cell layers, the outside, the endoderm and the ectoderm. Okay, so all this blue is mesoglia. They have something called a gastrovascular cavity, so it's just an opening where food comes in and then the digested stuff comes back out. So that doesn't go all the way through like the ctenophores. They have nerves, planula. That'll be important. Let's go over that in a little bit. But they are the larvae, and then nematocysts are the stinging cells, or they're the harpoons, not the stinging cells. So they all have stinging cells. That's how they capture their prey, and they eat, and they defend themselves. Okay, so this is a generalized life cycle of a cnidarian. Remember, there's 2 kinds of cnidarian body shapes. You have the medusa, which is the jellyfish, and then you have the polyp, which is like the little flower thing that looks like a coral. Some of them don't have both kinds, but this is the generalized. We'll start with the jellyfish, right? So you start with the jellyfish. You have a male jellyfish and a female jellyfish. They have sex. They release they have sex by releasing their their sperm and their eggs into the water column. They fertilize each other and they make a fertilized egg. What comes out of this egg is a planula. Remember this. planula is like a little flatworm moving thing that either like swims through the water or crawls around on the seafloor to find a place for that thing to develop into a polyp. Once it finds a nice place, it turns into this flower, and it stays like this flower forever. It never moves ever again. For the jellyfish, what comes out of this polyp, it just makes more of itself, right? This is budding reproduction. This is asexual reproduction down here. And it just shoots out medusas. Medusas basically are an inverted polyp. This is a polyp, this is a medusa. So a polyp is shooting out more of these medusa and becomes a medusa factory. And this is the life cycle of a jellyfish. That medusa then has sex and things starts all over again.
I don't know if I asked this before.
You did ask this before.
Yeah,
The polyp in jellyfish produces multiple jellyfish. Yeah. Mm-hmm. Oh, it depends on the species, but it produces multiple ones. So the next feature that is important for cnidarians, the cnidosite. Site means cell. Cnido, just the first part of cnidarian. This is the actual cell that produces the stinging tentacles. So the stinging tentacle itself is called a nematocyst. This is the cell that makes that sting tentacle. It shoots out water from this, shoots it out, shoots it into the prey, and that prey dies. Pretty simple. Okay? Going through the life cycles, the hydrozoans basically, so that life cycle is very generalized. That's, I guess, it's not generalized, it's specific for the jellyfish. The hydrozoans, they have a reduced medusa phase, or they don't have medusa phase at all. And the anthozoans, the corals and the anemones, definitely do not have any medusa phase at all. So they just go from a polyp, which has sex, and then makes an egg. That egg turns into a planula, it moves around, finds a spot to settle, and it makes a new polyp. So polyp to polyp. The hydrozoans are really, really good at budding off. So you'll see like they make these little buds, they bud off, and that is another version of this. It's just, it looks different. But budding usually refers to going from a polyp to another polyp, whereas budding in the medusa goes from, or in the jellyfish goes from a polyp to a medusa. But regardless, it's all asexual reproduction. You're just taking a little bit of yourself, throwing it out, turning it into a new organism. Okay, corals, last thing probably to note is that they have a relationship with dinoflagellates, which are algae that live inside of them, and they take in light to make fixed carbon photosynthate from CO2. Take CO2, paste together to make sugar. I don't know if they'll ask this twice on 2 different lab practicals, but it's a pretty important thing. All right. So that's that for this lecture. Let's take